All right, good afternoon, everyone. Today we'll be talking about the mechanobiology and biomechanics of the MPC. So as an introduction, the MPC is crucial for nucleocytoplasmic transport. It allows for the free diffusion of small molecules, along with the receptor-mediated transfer of large molecules. In this paper, we aim to discuss the structure of the MPC, the various transport models, and the alternate functions of MPC, including in gene expression. The MPC has many implications in health and disease. For instance, in cancer, there have been observed chromosomal translocation of knobs, and it has also been seen that inhibiting MPC selectively prevents tumor growth. Also, in neurodegenerative diseases, we have found out mutations in nucleoporins and knobs that are related to those diseases.
Few major structures constitute the MPC. A three-ring structure makes up the scaffold of the nuclear pore complex. The inner, outer, and membrane rings all have transmembrane domains that anchor the NPC to the nuclear envelope. The inner ring and the outer ring have similar assemblies called Y complexes, although the inner ring is a flexible structure capable of greater dilation and dynamism than the rigid outer ring. 8 cytoplasmic filaments extend from the cytoplasmic outer ring. Their function in nuclear translocation remains relatively unsolved, although these filaments interface with cargos in the cytoplasm and contain binding domains for import and export machinery. Mutation of these domains has little impact on MPC function. 8 nucleoplasmic filaments extend from the nucleoplasmic outer ring and are joined in a distal ring, creating a basket-like structure. The basket filaments are posited to contribute to NPC assembly at the internuclear membrane and contribute to the curvature of the nuclear envelope. The nuclear basket also interfaces with chromatin and may thus play a role in nuclear organization. The FG nups are intrinsically disordered proteins extending into the central channel of the NPC, with characteristic repeats of phenylalanine and glycine. Interactions between these IDPs likely dictate selective transport through the NPC.
There are various factors affecting MPC transport. For example, in small molecule diffusion, the KBT serves as the soft limit for transport. Particles larger than 5 nanometers are shown to have low probability of passing through. Additionally, this is due to the high energy penalty for large molecules since they need to push the high density nubs aside. The large molecules display more FG nubs compared to the small ones. For large molecules import and export, the nonspecific proteins increase KD, decrease binding strength between the nuclear transport receptors and the FG nubs. Also, in more densely grafted surfaces indicates more available NSP1 FG molecules, which indicates higher apparent binding affinity. Shape also plays a key factor in large molecule import and export. For instance, even a cylinder molecule has a larger volume than a spherical molecule. If the cylindrical molecule has a narrower radius than the sphere, then it'll pass through the nuclear pore complex more easily. Finally, for the RAN-GTP and RAN-GTP gradient, it plays a key role in the mechanism of the transport. So binding of RAN-GTP dissociates the import complex, while hydrolysis of RAN-GTP to RAN-GDP dissociates the export complex. The gradient is also very important for maintaining directionality, and previous studies show that disruption of the gradient reverses the direction of nuclear cytoplasmic transport.
So how does transport through the NPC occur? Multiple promising models exist defining the mechanism by which the FG nups enable bidirectional high-throughput diffusion of small molecules and NTR-bound cargo while repelling inert cargo. The selective phase model suggests that cohesive interactions between FG nups create a hydrogel-like structure that only interactions between NTR-bound cargo and the FG nups can disrupt. The virtual game model posits that non-cohesive interactions between FG nups create significant crowding within the central channel of the NPC, and that binding of NTR-bound cargo to the FG domains can overcome this entropic barrier. The forest model proposes two zones of transport, a peripheral non-cohesive zone where small molecule diffusion dominates, and a dynamic cohesive central zone where selective transport of large cargo occurs. None of these models, however, are definitive, and it remains unsolved how translocation through the NPC occurs.
So there are various alternative functions of the NPC. First of all, the NPC components can associate with specific genes and regulate their function in a transport-independent manner. Usually, the NPC organizes and regulates gene expression through several overlapping mechanisms. It involves modulation of the chromatin state and architecture, post-translational modification of chromatin-associated proteins or nubs themselves, along with tethering of specific transcription factors to control gene expression by the recruitment of respective DNA binding sites. Aside from silencing genes, NPC components can also act as promoters. For example, NUB210 added to the NPC during myogenesis is not necessary for transport, but is indispensable to regulation or turning on of the muscle genes.
In conclusion, the nuclear pore complex is one of the most efficient known biofilters, a complex protein structure with over 30 components in many copies. Understanding the mechanism by which thousands of nuclear translocations can occur per second in a highly selective and bidirectional manner has significant potential for biomimetic design. Harnessing the capacities of the NPC would allow engineers to develop high-throughput, semi-permeable barriers in mechanics and biology alike. The evolutionary develop and the NPC and its dysfunction and disease also create opportunities for innovation in biogenesis and medicine.
All right, thank you very much.